great honor to be able to speak with everyone tonight. So this presentation is meant to be the first in a series of 3 webinars. It's meant to go over primarily from a very high level, very sort of a cursory introduction to what does the normal single cell workflow look like? What are some things to think about when you're setting up a single cell experiment? And part 2 of this webinar series, it's in I think 2 weeks, maybe in a month. I'm not exactly sure on the timing. We'll cover more tips and tricks and more practical aspects of it. The legal disclaimer, of course, the QIAGEN products that we talk about are for research use only. They're not for diagnostics. They're not for treating any sort of diseases. I'm going to start with going over on a very introductory level why we should care about studying single cells. There's a few things I want to touch on. Some are very pragmatic, some are more, have to do more with analyzing biological samples. And then I'll go into the basic parts of a single cell workflow. These aren't too different from doing a normal bulk sample workflow, with the exception that you need somehow to go from a bulk sample down to a single cell. And there are a few methods to do that. I'll touch on those briefly. I'll cover whole transcriptome and whole genome amplification technologies. Most of the molecular biology protocols we have aren't meant to deal with the amount of DNA or RNA that's in a single cell. There are a few that are. Maybe you have an extremely sensitive PCR assay, for example, where you can do this directly on the DNA out of a single cell, a single genome's worth of DNA. But for the most part, it's usually necessary and quite often recommended to amplify the DNA somehow. After this, I'll touch briefly on analytical techniques. So maybe you do microarrays, maybe you do PCR, but this will depend very much on the research question that you're particularly interested in, as well as the data analysis will. At the end, I'll touch briefly on QIAGEN products for single cell analysis, and I'll focus there primarily on the whole genome or whole transcriptome amplification. If you have any questions regarding any of this presentation, feel free to type them in the chat box, and Lily will save them until the end. So the first reason to analyze single cells is a very pragmatic reason, and that's that maybe you only have one cell. Maybe you have more than one cell, but maybe your cells are extremely hard to get. Maybe you're dealing with a small tissue sample, a small biopsy. If you're dealing with an early blastomere or in vitro fertilized embryos, you can only remove one cell of these without damaging them. If you're dealing with microorganisms from environmental samples, maybe you want to get a whole genome, but you can't culture these. The vast majority of microorganisms have never been cultured. And if you go at it from this approach, it's painful. So, just a sec. There we go. Okay. The second reason that you may want to be looking at single cells versus doing a bulk analysis is that the genome is not the same from cell to cell. Genome heterogeneity and mosaicism is the norm. It's not an exception. Every cell is unique. Your average human has something like 27 trillion cells. These are only if we count the human cells in the body. If you start counting bacteria on the skin and digestive system, you come up with a number that's much higher, something, I think it's 90 or 100% more than that. So during all of these cell divisions, I mean, everyone starts off as one cell, right? And somewhere online, you're now at 37 trillion, and during that time, you've had to regenerate your skin every 2 weeks. You've had to regenerate your bones every several years. There's been a lot of cell division. Yeah. During cell division, you've copied a lot of DNA, and it's really arrogant to think that sometime along during all this process and all your years on this world that there has not been a single mistake that has not been corrected, that has not been not corrected. So a single persistent error. I was just reading in the news recently that, I think it was a paper from Harvard, where they're looking at neurons and they're finding something like 15 or 1700 unique single nucleotide variations. So this would be insertions or deletions, transitions, transversions in each neuron. Yeah. If we look at aneuploidy, something like 3% of adult liver cells are actually aneuploid. They're missing or have an extra copy of an entire chromosome. In elderly subjects, 4.5% of fibroblasts are also aneuploid. So these sort of things that we think about, okay, it's a person that has a genome. Well, not so much. A person has many genomes. That's only if you're looking at, of course, genetic variation, but you can also look at transcriptomic variation. The changes in a genome that one cell will have versus another can, of course, manifest as changes in a transcriptome. So, for example, you might have a single nucleotide polymorphism in a transcription binding factor site, or transcription factor binding site. This can then, of course, affect the transcriptome in that this transcript may not be expressed in one cell versus the other. This is a really interesting heat map. This is quite often how you'll see RNA Seek done on large amounts of single cells, the data visualized, where you have, I don't know in this case if this side is genes and this side is cells, or the other way around, because they've analyzed a lot of cells. But you can look at the transcriptomes of all of these different individual cells and you can cluster them. And you can see that there's different groups of, in this case it was neurons and neuronal cells, and you can find new classes of, I believe, oligodendrocyte in this case, based on their transcription profile. So you have cells that are in the same tissue that may look, from a very microscopic level, they may look the same, but they're doing very different, they have very different functions. If you add on top of this the epitranscriptomic inheritance, so the fact that you, as a cell, you do what you do because you've inherited a certain mRNA from the progenitor cell that you arose from, yeah, there's a lot of variation here. And it's a lot of variation that we've typically just bundled up and counted among the sample-to-sample variability. The final reason that I want to convince you that we should be analyzing single cells is that what we're usually interested in is the cell as a biological entity, and what we usually measure are populations of cells. So if we take two samples, any two samples here, they could be from the same treatment, they could be from different treatments, it doesn't really matter. What we usually do is we grind up these things, we measure some factor, in this case it's the number that's within these circles, and we come out with a mean. What we're usually measuring is this population mean. Now, if you take two biological samples, you'll get two numbers. In this case, they're very similar, they could also be very different, and you'll come up with a very low standard deviation. You'll find that these two populations look the same. But if you were to look at these 32 cells individually, you would see that there's probably three distinct populations in here. You have these two numbers here, which are a high responder maybe if this is a treatment. You have this intermediate phenotype, and then you have a huge sea of non-responders. This is the sort of variability that you can capture with single cell analysis that you cannot even begin to address with bulk samples. So there are a wide variety of applications for single cell analysis. Pretty much any molecular biology method you can think of that you can apply to a bulk sample, you can apply in some form to single cells. Really common ones are whole genome sequencing. If you want to detect variability from cell to cell in genome structure, either SNPs, indels, microsatellites. If you want to detect larger structural rearrangements, aneuploidy, copy number variation, or if you want to de novo sequence unidentified, new, and organisms that you couldn't necessarily culture. So small single cellular organisms that you could not otherwise get a larger amount of DNA from. This is all possible. Targeted resequencing, if you're interested not in the whole genome, but only a certain target set of genes or a region in the genome, and want to multiplex higher levels of samples, this is a good option. If you're using microarrays, the same microarrays you use with bulk samples, you can use with the amplified DNA from a single cell. And if you're interested in RNA Seek, so to detect variability in transcript abundance, to look at specific pathways, or to segregate different types of cells from one another, to detect new types of cells within a tissue, this is a good choice. And finally, if you're, there we are. If you're interested in QRT-PCR profiling, measuring the transcript abundance or the expression of a single gene or a single isoform or splice variant of a gene, you can also do this on a single cell. So, in general, the workflow that you'll follow when you're doing a single cell experiment is more or less the same as what you would do with a bulk experiment. There are a couple differences, and I'll spend more time talking about the differences than the similarities because I think they're more interesting. So with a general workflow, what you'll do is you'll get a primary sample. In this case, we're taking a tissue sample, a solid tissue sample out of a patient. And from there, you will go straight into molecular biology of some sort. Usually, this is a DNA or RNA extraction kit, followed by some purification kits, followed by maybe library preparation and sequencing, maybe run it over a qPCR panel, and then you'll analyze the data with some sort of software. Even if it's just Excel, it's still a type of software. Now, the primary differences between a normal bulk workflow and a single cell workflow are that after you obtain this primary sample, you need to somehow isolate single cells out of this. And there are a few ways of doing this. And the ways you do this will depend a lot on whether you're dealing with a solid or a liquid, so a cell suspension. After this, because most of the molecular biology methods we have aren't sensitive enough to be done on the amount of DNA or RNA in a single cell, we usually have to amplify this. This is done with either a whole genome or a whole transcriptome amplification. This brings the amount of DNA up to a level where standard molecular biology protocols can work on that. So when we talk about isolating single cells out of a tissue, there's a couple different scenarios. The first one is that your cells are in suspension. So you've either grown up a cell culture or you're dealing with, say, cells in a blood sample, something along those lines. And there are a few ways you can isolate single cells out of this. The first is a very sort of smash and grab method. It's to dilute these. So you'll run a serial dilution. You'll quantify the number of cells in your sample quite accurately, and then you'll scatter these over a microtiter plate. Now, the occupancy of each well in this microtiter plate will follow a Poisson distribution where you'll have a lot of cells that are empty. You'll have a fair number of cells that have a single cell in them. You'll have a fair number of wells that have 2 or more cells in them. The problem with this is that you'll have a lot of these wells that are completely empty where you'll get no analysis product for. And this can be fine if you have a really cheap molecular biology method, right? You certainly wouldn't want to do NGS sequencing on all of these wells. But this can be an option. It has the benefit for transcriptomic work that when you're diluting these cells, it's really easy. You're not necessarily changing the transcriptome too much. There are other methods that are much harder on this, on the transcriptome of the cell, but this one is fairly good that way. The second way to isolate a cell of interest out of a cell culture is micromanipulation. So you take a sample of your culture, look at it under a microscope, and use a very small pipette to pick out the cell you're interested in. This is extremely labor intensive. You do not want to do this on 5,000 or 10,000 cells. But you get a very good idea about the cell that you have. You can visually identify it. You can take pictures of it. You can stain it beforehand. And in the case where maybe this cell is something that's very hard to identify visually, that image recognition software can't help with, this is potentially a good option for you. The most common method for isolating single cells of a cell suspension is definitely FACS. So you have cells there, if they're not naturally fluorescent, You need to label them with something. Usually this is an antibody to something that you're interested in or this specific cell population is interested in, and it's fluorescent. And then you pass them across a laser with a detector, and where you see fluorescence, you sort these into one bin. Where you see no fluorescence, you throw them out. This is extremely fast. I think FACS will run something like 10,000 sorting events per second. It's extremely fast, but you need to have a huge number of cells as input. The sorter itself is also quite expensive, and the whole process will change the transcriptome a fair bit. So it's a better option if you're doing genomics work. Microfluidics, this is something like the Fluid IMC1. There's also other platforms available. This is essentially a FACS miniaturized into a very small chip. And it has some of the same benefits that you can do a large number of cells and also some of the same drawbacks. So it's also an expensive machine and you also need a high number of cells. So these are all fine and good if you're dealing with cells in suspension, but this is not always the case. In fact, quite a lot of the time you're dealing with solid tissues. Your options here are more limited. What the most common, I would say anyways, I don't have any data to back this up, but what I believe the most common method here is to dissociate your solid sample into cells in suspension. And you do this by mechanically disrupting them. So you basically take a your solid sample, you grind it up with some detergents, not too much detergent because you certainly don't want to solubilize the cell membrane, but a little detergent. And you disrupt this into some sort of slurry that you can then handle like a normal liquid cell, like a normal liquid cell suspension. Another option is laser capture microdissection. So if you take a section of your tissue, you can then isolate off single cells using a laser and an adhesive cover slip. So for liquid, and dealing with cells in suspension, you have a lot of different options. And with dealing with solid cells, you have two options. One is laser capture microdissection. The other one is to make your solid into a liquid. Something to think about when you're choosing a method of isolating single cells is what is the scope of my experiment and what is the necessary throughput. So if you're dealing with a system that has a huge amount of cell-to-cell variation, maybe a lot of biological variation as well, you may need to do more cells. So something like FACS is more appropriate than, say, serial dilution. So before you start, it's a good idea to sit down and think about how much cell-to-cell variation you can expect, what the biological variation in a bulk sample would be, and then also the technical variation of your method for the downstream molecular biology and data processing steps. Some other miscellaneous things to think about when you're deciding on a method of single cell isolation. Think about handling your cells. Think of them as very delicate things because in a way they are. They're used to being, if you're dealing with a solid sample, they're used to being surrounded by other cells. So think about having an uninterrupted cold chain, of going straight from, or maybe even having no cell storage at all, maybe of going straight from cell sorting into a lysis buffer, or straight from cell sorting into the downstream molecular biology reagents. You want to minimize storage time when possible. And the other thing to keep in mind is that when you're dealing with a single cell, you need to, by necessity, have smaller volumes. You can't have a single cell in 1 milliliter and expect to recover it later. Another thing to consider is that when you're dealing with single cells, a small amount of contamination can completely swamp your signal. So if you have contamination of 1 genome in a normal bulk experiment, you probably don't care because your signal is supplying then 1,000, 10,000 genomes to that. If you're dealing with a single cell, well, this is now 50% of your signal. So contamination can come from your sample. Maybe you get 2 cells in your well. Maybe you have some DNA hanging around on the outside of a cell. It can come from your laboratory. You need to be really careful about this and run your single cell experiments maybe somewhere a little more clean. You need to use UV and sodium hydroxide or whatever sort of decontamination protocols need to be in place. They should be quite stringent. And then the final place is that the reagents you use for the whole genome amplification, these can also carry DNA or cells into your reaction. So you need to be especially careful about what sort of buffers and what water you're using for these procedures. So after you've isolated the cells that you're interested in, you typically lyse them and then do a whole genome or whole transcriptome amplification to bring up the amount of DNA or RNA in those cells. These can be classified into two primary groups. These are either PCR-based. I won't go into them here. This is scheduled, I believe, for the next webinar. But this is things like Malbec and like degenerative oligoprimer PCR. And then there's also PCR-free methods, such as the multiple displacement amplification, the MDA. After this, you can basically apply whatever molecular biology method you're interested in. It can be things like simple PCR of a gene of interest and then Sanger sequencing. This can be different qPCR. Either this is done on an RNA level or on a cDNA level or on a genomic DNA level to detect specific point mutations, specific transcripts or splice junctions or aneuploidy structural rearrangements, all sorts of things. If you're doing microarrays, that's also an option, and NGS as well. That's one I think a lot of us are using. And then data analysis and interpretation. I can't possibly to start to approach this subject on such a wide variety of molecular biology methods, but I will say that at QIAGEN we have tools for the analysis, particularly of next generation sequencing data, either whole genome or whole transcriptome data, and we have this is done with either the genomics or the biomedical workbench with CLC. And then downstream we have the Ingenuity that has a variant analysis and a pathway analysis pipeline. And these are quite useful if you're trying to make sense of transcriptome, whole transcriptome data done with a large number of data points, which is typically what you'll get out of a single cell experiment. So that's sort of a quick run through of the workflow. You'll obtain your primary sample like you normally would. You'll have to somehow get single cells out of this. You'll need to amplify either the whole genome or the whole transcriptome. And then after that point, you've essentially banked all of the genetic information that was in that cell. From there, you can go into one or more molecular biology steps plus data processing. And since you've amplified this DNA, you can then go back and do this perhaps a second time if you're interested in something new. And at QIAGEN, we have products for basically every stage of this process. For cell isolation, we have a platform which we have not introduced but are coming out with sort of in the middle of next year. I believe it's sort of April, May, June-ish, somewhere in there. For the amplification of DNA or RNA out of a single cell, we have the REPLIg portfolio. This is an MDA-based, a PCR-free MDA-based amplification method that can bring up the amount of DNA from a single cell to a point where you can use any of a number of molecular biology techniques on it. For the analytical techniques, we have DNA and RNA Seq library preparation kits. We have targeted panels for targeted resequencing, qPCR arrays. There's a huge volume of products we have there. And then for data analysis, we have the CLC bioinformatics software. We have qPCR analysis software. We have the Ingenuity variant and data and pathway analysis software. There's a lot of options here. So I'll take the third part of this seminar here and take, I guess, 10 minutes here to talk a little bit about our MDA technology and why this is so important for a single cell workflow. So this first step where you're essentially copying the DNA out of a single cell and using this to then do whatever downstream process, and then you're using this also to bank as a sort of a biobank. This is an extremely important process. This needs to be error-free, and it needs to have as much fidelity as possible. So it needs to be a true and accurate representation of the genome that you're interested in, the genome of that single cell. Because once you've done anything to this single cell, you can't go back and measure it again. This is sort of the curse of it. It's once you've measured it, it's gone. It's sort of a Schrodinger's cat type thing. So MDA, this could be any MDA, but it works by annealing random primers, short random primers, to a strand. This could be a first or second strand cDNA, or it could be a gDNA, so a long strand, a long chromosome. We have then a DNA polymerase. This is a variant of the 529 polymerase. It's not a thermostable polymerase. It runs at 30 degrees Celsius. It's a low temperature polymerase that is extremely processive. So it can, well, it prefers and it can and it likes to produce extremely long fragments, starting upwards of 2,000 bases up to 70,000 bases and even on occasion longer. And this will, of course, we anneal the primers at a low temperature, and at the same temperature, all in one reaction, without any thermocycling, the polymerase starts to polymerize here. And the other thing that is interesting about this polymerase, firstly, is that it's very processive. Secondly, is that it has a really high strand displacement activity. So when this polymerase runs into another primer, it will kick this strand off and continue polymerizing. Yeah? So you can end up spinning off multiple copies of the same strand of DNA all at the same time without thermocycling and without PCR. The third fact about, I guess, this polymerase, which is makes it really good for this application, is that it has an extremely strong proofreading activity. It's about 1,000-fold higher than TAC, which is, of course, not very impressive because TAC is, I think it's 10 to the 5th. But it's also, depending on which enzyme you're using, it's somewhere between 10 and 50 times better as far as not introducing indels and SNPs, or point mutations, than a lot of the high-performance thermostable polymerases that are out there. The final thing about this polymerase, sort of, I guess I've already touched on this, but because it is so processive, it has very little sequence bias. And what I mean here is that if there's any sort of secondary or tertiary structures in your genomic DNA or in your cDNA, this enzyme really doesn't care. It will read through almost anything without slipping, without pausing, without dissociating. So this produces very long fragments. As I said before, we can run this natively on genomic DNA, or we have a modified protocol where we can produce a first or second strand cDNA, ligate these together, and then run a WTA on that, so a whole transcriptome amplification. We have a variety of products. Some of them are specific for NGS. These are the REPLIg single-cell DNA and RNA library prep kits. These are the PCR-free MDA protocol we have for whole genome and whole transcriptome amplification, coupled with the PCR-free library prep. So you can go from a single cell all the way up to an NGS library within a single day without using any PCR whatsoever. So there's no need after this to, for example, deal with optical duplicates or PCR replicates. This is not an issue. This just doesn't happen. If you're not doing NGS, we have just the MDA section of this for either DNA or for RNA. And this can be used with a variety of downstream processes for microarrays, for, again, maybe you have a different NGS library prep you want to use, you can do that. You can run it through qPCR panels. And then a final set of products that I'd like to touch on briefly is not really single cell, but it's sort of a small input or very low number of cell platform. And that's with a small number of cells. In this case, it's 25 plus. So if you have 25, if you're able to come up with 25, 30, 50 cells, something along these lines, you can analyze the whole transcriptome and the whole genome in parallel. Now, this would be the REPLIg WG and WTA kit. Some of the features that make the REPLIg kits really good for single-cell analysis. The first one is that there's minimal background. We have taken extreme care during the manufacturing process to eliminate any chance of contamination. These reagents undergo some special treatments that are proprietary here, where this completely degrades any sort of DNA or cells that could be contaminating. So in this experiment, we take 2,000 copies of a bacterial genome, which is, I hope, more than you would ever expect to contaminate your cells with, your single cell with in a single cell experiment. We can spike this in before the treatment, and with various qPCR assays, we cannot detect this afterwards at all. This is 2,000 copies, so it was quite a bit. The REPLIg single cell kits, they produce a lot of DNA. It's more than enough DNA for most downstream applications. They generally produce at a bare minimum 10 micrograms. This is with the Parallel kit here. But typically, we expect somewhere between 30 and 60 micrograms of DNA from either whole transcriptome or whole genome. Now, this is a huge amount. It's more than you'll need for most analyses. And it's enough to do something like a whole genome sequencing experiment or a whole transcriptome sequencing experiment and still have enough left as a sort of a bank, a biobank, in case you need to go back to that cell and do things later. The library prep kits, where we've integrated the MDA plus the PCR-free DNA or RNA Seek library prep, these produce more than enough library for sequencing once, twice, 3 times on a HiSeq or a MiSeq or an XSeq. Yeah, so we offer them on their own as just the whole genome or whole transcriptome application, but then we've also integrated these with completely PCR-free NGS library preparation protocols, where these are two tube protocols. Typically, we do the whole genome amplification or the whole transcriptome amplification in one tube. This takes a few hours in all cases. It's sort of the longer part of this protocol. And then after shearing the DNA, we can go into end repair, A-tailing, adapter ligation, and cleanup. These kits are offered as a single package. Whether you're doing DNA or RNA Seek, you can go from a single cell, including isolation, all the way up to a PCR-free NGS library within a day. The other thing that all of these kits offer is even coverage. So what I mean by this is that when we're amplifying a copy of the DNA, we're not missing parts of it. We're not over-amplifying and we're not under-amplifying parts because they're, say, GC-rich or hard to copy. What this means is that you'll have fewer dropouts, so fewer loci will, or I guess the other way to look at it is more of your loci will be present in all of the cells you're looking at. This is a good thing no matter if you're doing NGS or different analysis. If we compare in this experiment, this is a continuation of the previous experiment, where we sequenced a bacterial genome, 1 picogram of bacterial DNA, with REPLIg and with a PCR-based competitor using the same NGS library preparation protocol. So we've controlled for everything possible otherwise. We see a really even coverage with the REPLIg and sort of a very poor coverage of low GC content regions with the PCR-based method. Now, when you look at this data, this is a pile-up just from the BAM files. We see a lot more spikiness in a way here, for lack of a better scientific word. And in certain cases, you can say, okay, well, that would be great if my gene of interest is right here, right, where I have this 3,000 reads piled up over top of it, this massive read depth. But the other thing is that what you don't see here are a lot of very small missed regions where we just don't have any data at all with the PCR-based competitor. Now, that's fine if you're dealing with a genome experiment, a whole genome sequencing experiment, but when you're talking about RNA Seek, this deals directly with the number of transcripts that you're going to detect. Yeah? So the more even your whole transcriptome amplification, the higher number of transcripts that you're able to copy into DNA and then feed into a library prep or feed into a qPCR assay. So this deals directly with the number of transcripts you can detect after. So with this experiment, we took 50 cells, did an NGS, RNA Seek library prep, then 25, then 10, 3 several times, 1 several times. And of course, the number of genes you detect goes down as you go down to single cell. In a complex tissue, I do not expect every single cell to be expressing the exact same set of genes. I expect that there's the vast majority of genes are shared and common, but I expect there to be also differences, so expression that's unique to a single cell. And if we take just any three single cells, we see that this is the case. We have a vast majority. In this case, it's sort of two-thirds upwards, depending on which library you're talking about, which libraries you're comparing, that are shared between cells. But on top of that, there's also a lot of genes which are expressed uniquely in one cell or a different cell. Yeah? And when you take the sum of all of these, you approach the number that you would get in a bulk sample. So what you're able to do here is you can say, okay, in a bulk sample, I have 10,000 genes that I'm able to detect, yeah, and quantify. And with single cells, I can get maybe 7 or 8,000, and 7 or 8,000 in another cell, and 7 or 8,000 in another cell. But if you take the total number of genes that you're able to quantify, you're back up at that level. So we're not losing sensitivity by doing single cell sequencing. Far from it. We're able to partition out this variability into different cells from a whole tissue level. Going back to the fidelity and the proofreading activity of this polymerase, this is really important if you're interested in any sort of single nucleotide variation, because this directly contributes to the amount of background you'll have to deal with. So with more background, you need to set much more aggressive cutoffs and thresholds as far as what you're interested in. So for example, if I see single nucleotide variations in all sorts of reads, well, if I see a single nucleotide polymorphism in the gene that I'm actually interested in at a low proportion of the total reads, I can't distinguish that from the background, so I don't know whether that's interesting or not. So as I mentioned before, this polymerase has a really good proofreading activity. It's much higher than TAC, and it's much higher than any of the sort of high-performance thermostable DNA polymerases that are on the market today. Furthermore, because we don't have any PCR, any misincorporation, that's sort of a dead molecule. It's not copied again through PCR. So if the polymerase misincorporates a nucleotide, of course the proofreading activity is better, so there's a good chance that it fixes this problem. But if it doesn't, this molecule that it just made and this error that it just introduced isn't copied in the next round of PCR because there is no next round of PCR. If we look at NGS data, this is on top, done with REPLIg, and the bottom with a PCR-based approach, we see that there are a lot of sites here that aren't matching the reference genome. And a lot of these are spurious. They're not knowing the actual sample. We don't believe these are coming from actual cell-to-cell variability. Depending on how you're doing your PCR-based method, if you're using a really good high-performance polymerase to do the PCR, you may get 10 times more errors. In this example, we see 40 times more errors with the PCR-based method than with REPLIg. And this is at similar sequencing depth, similar percentage of mapped reads. So if you're doing single nucleotide variation detection, you definitely want to pay attention to the method you're using for whole genome or whole transcriptome amplification. So this is a very brief overview, and I haven't been able to impart too much information, but what I hope I've been able to convey is that there are a few reasons that you should be analyzing single cells over bulk samples. The first is very pragmatic. So if you only have a single cell, you don't have a choice. Yeah? This is what you're stuck with. And you can either move on to a new experimental area, or you can put in the extra effort and you have to do a single cell analysis. The genome and transcriptome are different for each cell. And what we've typically done is analyze populations of cells. And what we need to do moving forward is really interrogate each single cell individually, because these have very interesting effects. When you're setting up a single cell workflow, there are a few things you need to consider. For the most part, the molecular biology and data analysis that you do downstream will be very similar to as if you were doing a bulk sample. What you need to give extra attention and extra care to are the method of isolating single cells, especially if you're coming from a solid tissue where tissue disruption may extremely change the transcriptome profile of your cells, and the method that you're using to amplify the DNA or RNA out of that, because this is setting the stage. After this point, if you've screwed this up, there's nothing you can do to rescue it downstream, because that cell is gone. QIAGEN, we have a wide variety of products for all parts of this workflow, including soon the single cell isolation. And a lot of these applications are enabled by REPLIg, by our PCR-free multiple displacement amplification. This can be used on either DNA or RNA and offers minimum background due to our unique manufacturing process. It produces a large amount of DNA. This is enough to run whatever sort of molecular biology protocols you're interested in, as well as to bank for future use in a way to, because you can't save the single cell, but you could maybe save a copy of the DNA from the single cell, sort of the next best thing. We've integrated them with a PCR-free NGS library prep, which means that you can go from a single cell to PCR-free NGS libraries within a day. And then because of the characteristics of our polymerase, because of the high processivity, because of the high proofreading activity, we get even coverage, which manifests either as better genome assemblies, fewer dropouts if you're looking at single loci, or better transcript detection, as well as fewer sequencing errors. With that, I think I will give control back to Lily, and we can maybe have a short Q&A session. So thank you very much for your time.